Top, it's so great to be here. Um, Thanks for breaking away from your busy schedules and other distractions. And I'm really excited to talk about one of my favorite topics, which is seeds, nature's perfect package. And what I did today is I put together a talk that just starts a little bit with the evolutionary history of seeds. And then we'll talk a little bit about the structure of seeds and what the functions of some of those structures are, and then the evolutionary adaptations of seeds that really we take a big advantage of in terms of plant conservation. So I'll talk about how some of the traits of seeds are really optimal and make plant conservation much easier than they are, say, for animals. So to start our talk, and let me just advance here, we're going to go all the way back to the Devonian period, which was about 400 million years ago. And during the Devonian period, it was a very much warmer time on Earth than it is today. We only had spore-bearing plants at the start of the Devonian period. And this was a period in which we saw a lot of radiation, not only of, evolutionary development of a lot of different types of fish, but also a lot of development in plants. So we had plants really expanding their colonization of land. We see the earliest leaves and true roots from this period of time in the fossil history. And we also see the first lignin appear and then the first trees. So there was a lot of expansion of speciation happening during this period. Just about by the end of the Devonian period, we had large forests developed across the land, and it's during this period that we see the earliest undisputed seeds in the fossil records from several different taxa, some from some sites in West Virginia and Wisconsin, and also from, I think, Belgium. So the plants that we see these early seeds on are called pteridosperms, or seed ferns. And what we see is that these were not true ferns. They had fern-like leaves, just to be clear. On these plants, what we see is some very early naked and seeds that are really not attached to the plant. So they're just loosely held in a structure on the plants. So that's the earliest seeds. And then after this period, we go into the Carboniferous period, which was up to about 300 million years ago. And during the Carboniferous phase, or the coal age, we see conifers appear. And by the middle of this period, we start to see the earth start cooling and drying. And by the end of the Carboniferous period, we see the collapse of a lot of the tropical forest. And this is when we really start to see the seed-bearing plants become much more dominant as opposed to the spore-bearing plants. So why did that happen? And for that, I thought it would be interesting to take a look at the spore-bearing plants' reproduction and what happened to form the seed. So I'm taking you a little bit back to high school biology here. And if you remember from high school biology, plants exhibit alteration of generations. And just wanted to, for those of you who haven't thought about this in a long time, just want to remind you how plants reproduce. So this is a typical cycle of a spore-bearing plant, and we're using a fern as our model organism here. And the fern, through meiosis, and meiosis is when cells split and the two child cells of the splitting cell each get a half of the copies of the produced spores in that process, and these plants would produce and release the spores that could be carried by wind. The spores would land on a suitable site and then develop into a gametophyte. And a gametophyte, again, only has one copy of chromosomes. And if you are ever, you can see these gametophytes. For ferns, I've seen them in some of our west side forest. You're in a much drier site in eastern Washington, but we can see these gametophytes on some of our damp logs here in our forest. And these gametophytes in structures called Anthroduridia and Archegonia will, through cell division, regular cell division, where each child cell gets a full copy of the one copy of the chromosomes, they will produce both sperm and eggs on these gametophytes. The sperm will need water to swim to the egg and fertilize the egg. And then, because you're combining a sperm with one copy of a chromosome and an egg with one copy of this chromosome, you then get a zygote that has two copies of the chromosomes. And that is what develops into what we are commonly familiar with is the fern, which is what we call a sporophyte. It has two copies of the chromosome. And so I remember when I first learned about alternation of generations, I was kind of blown away, like, oh my gosh, there's a whole different life form that this species has, that it alternates every generation. I just thought that was really fascinating about plants, and I continue to really be interested in that. So note here that in order for this reproductive cycle to complete, you have to have water because are swimming to the egg. So we know that's not true anymore. And in fact, it was some of the evolution of the seed as well as the spore into the pollen grain that helped seed plants evolve and become dominant. What we see in the fossil record is that the spore-bearing plants started producing two different types of spores, megaspore, what they eventually called megaspores and megagametophytes. So they started differentiating, and the megagametophyte would eventually turn into the gametophyte that produced eggs, and the megaspore would produce the sperm. And what we then see in the fossil record is that the megagametophyte, the female version of this part of the cycle, was retained on the sporophyte, or on this diploid individual. And it was retained in a structure called a cupule. So then, and it never left. So it would grow right there on the sporophyte and then would get fertilized by the megaspore. And of course, then if the megaspore landed right there with the megagametophyte, the reproductive cycle just happens right there on the sporophyte. Well, that cupule ultimately became the ovule of a plant. And then that allowed all the fertilization to happen right there on the plant. And eventually we see the evolution and the development of the encasing tissue that ultimately, after fertilization happens, some of the encasing tissue develops and starts forming a hard seed coat, and eventually the seed. And concurrent to this, so instead of this whole life cycle happening independent and out in nature in potentially inhospitable places, the life cycle now is happening completely on the sporophyte. So the whole part of this cycle that's in blue is now happening right within this, this structure on the plant. And that ultimately became the ovule. So there was a huge advantage during the late Carboniferous period, when things were really drying out and becoming colder. We had now a protective site for these female gametophytes. We had weren't reliant on water any longer, and we also then had the formation of a very hard seed coat that enabled these zygotes, these seeds, to survive some very harsh conditions. And so that's really sort of that is the evolutionary advantage that seed-bearing plants had, and why we see that they are so predominant in our flora today. Hold on. Okay, so let's take a look a little bit closer as to what structures of the seed are. So basically what we're looking at is we're looking at an developed or undeveloped to developed embryo in a package with a seed coat and a food supply. So I saw this quote somewhere, it's an embryo in a picnic basket. And this is also something that gave seeds a big evolutionary advantage because they have some of the initial resources they need when they land on a site. So they have an advantage getting established initially compared to the spore-bearing plants, yet another evolutionary adaptation. I show here a drawing that has both the monocot and dicot. So the bean is representing a dicot and typical embryo. You have inside of a seed, you have the early cotyledons as well as the early root radical here. So that's the embryo right there with what we call plumules, which are the leaves, and then the hypocotyl, which is the root radical. And then you have in a bean, this is actually a bean, we have this huge food supply, which the cotyledons that go with the seed, and then a seed coat. In monocots, we have, and some other seeds, we have an endosperm, and the endosperm is the food supply. But we also still have the embryo right here, down in here, with a root radical and the early leaves. So very similar structures, but the way everything is laid out in seeds varies depending on what family, what type of seed we're talking about. And in fact, we can loosely classify 12 different types of seeds in terms of their physical layout when we're looking at seeds. Not that important what they all are, but just know that there's a lot of variation in how those structures lay out. And then also with, for instance, if you're looking at an orchid seed, which is tiny, the food, there may not be hardly any or no food supply, it's only a few cells, and it's an undifferentiated embryo. So it can be very variable, but this is typical for a lot of species. So let's take a look at the seed coat. If you haven't ever had a chance to look at a seed coat through a microscope, I really recommend it. They're beautiful. There's a lot of variation in the structures of the seed coat. There's a lot of functions for the seed coat. They provide chemical and physical protection for the seed. So the seeds can have chemical compounds that are toxic to animals and seed predators. They can also have a lot of different types of structures that you see a variety here that can also hinder predation by animals. But those structures can also vary the movement of water and air across the seed coat. Some seed coats can be completely impermeable to air and water movement, providing some real protection against desiccation and whatnot. But others are breathable and do allow some uptake of water. Let's see, sometimes we see parts of the seed coat that can help with dispersal, and I'll get back to that. So all the different types of structures we see are ecological adaptations. So they might differ based on the climatic conditions where the species occur. They may vary based on the dispersal mechanism or the anti-herbivory adaptations. Let's take a look at some of the dispersal mechanisms that plants use. As we know, plants can't move themselves, and so they rely on dispersal of their progeny to migrate. Some of these mechanisms allow for very long distance migration, and others allow for almost no distance for migration. So air is really common mechanism. So here we see the sort of the hairy styles that develop after seed maturation on some of the Rosaceae family that I'm sure you're familiar with. So those will be picked up. Typically what we see is right at seed maturation, the seed becomes loose and breaks apart evenly and very easily at this point, and a puff of wind can move these on. We have water down here in the lower left. So this is an African dream herb, and it relies on water flotation to disperse its seeds. And we have a very hard external seed coat. And then there's usually air to make these types of seeds buoyant. So there's air entrapped within the seed coat. These can travel, these will actually drop into the ocean near the southern tip of Africa and travel for thousands of miles. So it's a really effective dispersal mechanism. Mechanical is another dispersal mechanism that we see. So we sometimes refer to these as poppers, and I think some good examples is some violets will do this, some phlox will do it. I think this might be corydalus seed, I'm not sure. And so this is relying on tension created by water or humidity that can build up and burst at the right time. But as we all know who have tried this, sometimes actually pressure, touch can also set these off. And so when you're trying to collect the seeds, you got to be ready for them to pop in your hand. And then animals provide a lot of mechanisms for dispersal. The classic one that has been talked about quite a bit is the chili pepper. And the chili pepper is a great adaptation, evolutionary adaptation, because the chili pepper produces capsaicin, and birds don't have the gene or the receptor to be affected by capsaicin. So they just think chili peppers are super yummy and gulp them down. And the seeds pass right through their system and then get dispersed when the bird excretes it. So it's a great evolutionary adaptation. Capsaicin is actually really energetically demanding. So there's always variation in the amount of hotness or amount of capsaicin in chili peppers. And it's thought that any population would have both low and high levels of capsaicin to help these populations with dispersal, but also during years in which the harder years in which there's drought or poor conditions. And then we have seeds that have a seed coat with hooks or different ways to grab onto a passing animal or a passing pant leg or shoestring, as we've all seen. And this can be a really effective method for traveling some distance. And then many fruits or many seeds are encapsulated in very tasty berries to attract animals to eat them for dispersal. Okay, so let me turn my attention to another way we can look at seeds. And this has to do with to what extent that seeds tolerate desiccation. And desiccation is another evolutionary adaptation we see in seeds from typically from the colder climates of the world, the northern hemisphere and the southern part of the southern hemisphere, to allow the seeds to survive cold, dry, harsh winters without dying, because they'll go typically through being desiccated as they're going through winter. So it allows them to survive that winter and then be ready to germinate in the spring. Seeds that can tolerate desiccation we call orthodox seeds. And it turns out in our flora in Washington state, we have mostly orthodox seeds. And this is really fortunate for conservation purposes because it's very easy to handle these seeds and store them for long periods of time. On the other hand, there are seeds that don't tolerate desiccation, and those are called recalcitrant seeds. And these are seeds that will just die. The embryos just cannot survive very low water content that you would see during the desiccation process. This is typically our tropical species. Some of our species that are ancestors or descendants of tropical species. So there's certain maples, for instance, in our flora that actually are have intermediate levels of tolerance for desiccation. A variety of other species. We also see recalcitrant seeds in some of our temperate rainforest species, like Curtilus will be desiccation intolerant. And a good indication that you have an intolerant seed is that when the seed disperses, it's still green and soft. And so that's a good hint that you might have a recalcitrant seed. Intermediate seeds are seeds that fall somewhere on the scale between orthodox and recalcitrant. So orthodox seeds, we can use very basic drying and freezing to store these seeds, which is just fabulous for conservation purposes. Recalcitrant seeds, we cannot do that, and it requires special handling techniques. Recalcitrant seeds cannot tolerate desiccation because of some of the materials, the storage materials in them are really subject to degradation because they have a lot of chemicals that are easily oxidized or peroxidized and break down very fast. Typically plants or seeds that have a lot of lipids in them are going to be, and which tend to be some of the larger seeds, like an avocado is a great example. These are recalcitrant seeds. And we will typically use cryopreservation, which is what I have this picture of, is a liquid nitrogen tank. And we will typically have to figure out what the phase transition of the lipids are. It's very technical. I'm not going to go into it because it's not my specialty, but we do look at how the very specific chemical properties, what chemicals make up the storage properties and what their phase transition is, and then figure out how to store them. This is something that's done a lot with the Hawaiian species that are highly imperiled. We do a lot of banking with them in a liquid nitrogen tank. And I will just point out, this is Dr. Christine Walters at the National Lab in Fort Collins, Colorado. And she has done a tremendous amount of work on understanding seed physiology and storage properties. So whenever we have questions, we go to her. There are a bunch of seed banks across the United States. Many of these are at botanic gardens that are a part of the Center for Plant Conservation Network. The Miller Seed Vault is the one we have at the University of Washington. That's us, which I'll talk about a little bit more. This one in Fort Collins, Colorado, is the National Center for Genetic Resources Preservation, the picture I just showed you. And that is the facility that where our food supply is backed up. So just as a little detour here, when I first visited this facility, I was very impressed that they have stored. So the room what we're looking at right here is a freezer. This is actually an entire huge warehouse room that's negative 18 degrees Celsius. You wouldn't know it by the fact that we aren't wearing jackets, but we weren't in there for very long. And what we're seeing on all these shelves are seed packets of every type of crop, every variation of variety of every type of crop that we eat in our food supply. So it's really a pretty amazing facility. And that's at the biggest scale that we do seed banking here in the United States. That is predominantly agricultural species, but each of the seed banks devoted to native plant conservation, like the Miller Seed Vault that I manage, we also send our seeds to back up here at the National Lab. So they're a partner for us, and we send our most imperiled native species also to this facility. Our Miller Seed Vault is at the Center for Urban Horticulture in Seattle, for those of you who've familiar with our site. Our seed bank is held in this commercial grade freezer. It's negative 18 degrees. This processing room that we're looking at is climate controlled at 15 degrees Celsius, which is about 57 degrees Fahrenheit, and 25 to 28% relative humidity. And we'll bring seeds into that room and store them for about a month, and that'll equilibrate the water content of seeds down to about 6 to 8%. So it super dries the seeds. And the work by Dr. Walters shows that that is optimal. That is the way to optimize your shelf life of seeds. We don't know how long of a shelf life we'll get on these. It could be for decade. I know it'll be for decades. It could be for centuries for some of our species, but we'll see as we go, because we've only been seed banking this way for a few decades. We currently have 142 species of Washington rare plants in the seed bank, and over 800,000 seeds. And again, each seed is an individual. So this is why we seed bank. This is a really inexpensive way to assure longevity and conservation, what we call ex situ or offsite conservation of our flora. And we try to collect from all the sensitive species, and we also collect from multiple populations. So we capture the genetic diversity of our native flora. Let me tell you a little bit about how we do our collections and how we handle them. So what we have on the left is how a typical collection is made for conservation purposes. We use coin envelopes, really handy. We're usually making very small collections because we're working primarily with rare plants at the Miller Seed Vault. Each coin envelope holds the seeds from one maternal line. So we know all our seeds in each envelope are genetically related. We have volunteers who work in the seed vault to hand clean all of those seeds. Again, our collections are typically very small because we don't want to impact the wild populations. So we separate the seeds from the chaff and package them up into these glassine envelopes. And then this whole collection, which represents one population of the species, will get packaged up into one or two of these aluminum sealed envelopes, which will then protect they're airtight and they're waterproof. So when we put them in the freezer, which is a fairly damp environment, it'll preserve the dryness of the processing room for those seeds, and they go into the seed vault like that. And then we can take them out as needed to do some testing or to provide the seeds for reintroductions or research. So I'd like to also turn to another aspect of seed conservation, and that is the germination and viability testing of seeds. And to do that, we have to understand a little bit about seed dormancy. And so getting back to the sort of central theme of this talk, which is the evolutionary properties of seeds, dormancy is another really important evolutionary property that we want to understand if we're working with these species. So let me give you a little bit of background about dormancy and what type we see, and then how we take that into consideration as we do germination testing. So we have generally two types of dormancy. We have those that are controlled by the embryo, which we call endogenous dormancy. And in that group, we have two subtypes. We have physiological dormancy and morphological dormancy. And physiological dormancy is most common. And that's when there's physiological changes in the embryo that need to happen before embryo will be ready to germinate. Morphological is when you see underdeveloped or non-differentiated embryos. So these are typical in orchids, also some ericaceous species, and I see it in some ranunculaceae, the buttercups, and they require pretty special handling when you're working with underdeveloped or non-differentiated embryos. They're a little bit harder to work with. So the second big group of type of dormancy is what we call exogenous dormancy, and this is what is controlled by the characteristics of the structures of the seed. So it can be a physical dormancy, like a hard, impenetrable seed coat that we would see in legumes that don't allow water to cross the seed coat. It could be chemical, where we see actual chemical inhibitors in some of the structures. We see that in a variety of families. And then it can be mechanical, a hard woody fruit, like in the walnut family. So there's a lot of things that actually control dormancy in plants, and I want to talk about what those are. A lot of tests have been done to figure out these mechanisms, and it turns out that that environmental conditions that are apparent while the seed is developing can have a pretty interesting effect on dormancy. So the amount of carbon dioxide, for instance, that's available, the amount of oxygen that's available, the length of day during seed maturation, the light quality, the length of the spring growing season, soil and mineral nutrition. These can all be environmental controls on dormancy and can even affect within population dormancy. But on top of that, we can have a hereditary component to dormancy. So we can have hereditary variation across populations. For instance, I think the lodgepole pine is a great example of this, where what in Western Washington is called the shore pine doesn't need fire to break dormancy, and then the lodgepole pine that is in fire adaptive systems do need fire. That's a great example of a hereditary population level hereditary factor. But also it can be based on the maternal or paternal plant. So a good example is lettuce, Latuca sativa, and this is a study that was done early on, and they determined that the ability of the seeds from this lettuce species to germinate in darkness at 25 degrees C is actually controlled by a gene from the pollen parent. So it's a father parent that affects this ability. Conversely, we have an example here of an effect by the maternal parent, and I love this story. This is the South African ice plant. It grows in mats, and it produces capsules both in the center of the plant as well as around the periphery. And it turns out that the capsules in the center are bigger and have about 200 seeds, and the peripheral capsules are smaller and have about 125 seeds, but the peripheral capsules, the seeds in those have higher germination rates, about 80%, when held for about 18 days at just normal temperatures, room temperature, but only 20% germination for the seeds from the central capsules. So some real variation just based on where the capsules are on the plant. It turns out that the peripheral capsules on the edge of the plants will easily dehisce from the plant and tumble and get carried and dispersed by the wind. So these are colonizing capsules. Seeds are smaller, lighter, and will be more easily to germinate when they land in a suitable place. The central capsules are retained on the plants, and those seeds are going to fall right beneath the plant. and they're going to hang out there in the seed bank, and potentially when this parent dies away, those seeds might germinate and replace the parent. So it's a great example of some maternal effects that you can see on dormancy. Other effects that you can see on the maternal plant is the physiological age of the parent. Also the seed, the position of the seed within a fruit. So for instance, seeds that are closer to the calyx end of a fruit might be have a different properties or dormancy than say ones that are out at the tip that are immediately released. All cool stuff. It's also really pretty amazing in the variation. The other thing to keep in mind with dormancy is that it's not a static condition. So I have a little diagram here that shows that when the seeds are shed from the plant, they might shed either in a condition in which they're dormant and they will not germinate under any conditions, or they're non-dormant, in which they'll germinate under a wide range of conditions. But that's not a static effect. It can actually, over the season, change and go from being non-dormant to conditionally dormant to dormant and back again, depending on environmental conditions. Let me give you an illustration of this. This was a study that was done with common knotgrass, Polygonum aviculare. And let me introduce you a bit to this very confusing graph. So what we have on the x-axis is time, starting with the month of October and going for about 20 months. And then on the top graph, we have percent germination. And then the bottom graph, we have days to 50% germination. And so what they did in this study is they took packets of seeds and they buried them at the site. And once a month, they dug up the seeds and then sowed them at 4 different temperatures listed here, 4, 8, 12, and 23 degrees Celsius, and sowed them for a period of time to see what the germination rate is. So when they buried all the packets in October, and the first month or two, in November, December, they dug up some seeds, sowed them, and what they see is that only the seeds at 4 degrees, sowed at 4 degrees Celsius, which is what we'd call cold stratification, that's where they got 80% germination. But it took, if we go down to this graph at the bottom, it took around 100 days to get that 50% germination on those seeds. Conversely, what the seeds are sown at the higher temperatures had very low germination rates when they were dug up in December, November, December, about less than 20%. And then they kept on repeating this experiment each month. So by February or early March, which is about here on this graph, they dig up another packet of seeds, sow them at these different temperatures, and now they're getting much higher germination rates at every temperature that they're sowing these seeds. And that is showing how these seeds are now conditionally dormant. Given the right temperature and the right conditions, they're no longer in deep dormancy, they're conditionally or non-dormant. So we're getting much higher germination rate. And in the 4 degrees C treatment, if we go down to this bottom graph, what we see is it only takes 20 days in this situation to reach 50% germination. So many of the seeds are now non-dormant or very lightly conditional dormancy. But by the time we get to summer, June, July, August, in fact, now when we dig up those seeds and run our germination test, these 3 higher temperature treatments are no longer getting good germination. These seeds are dormant again. So it's only the seeds that are given the cold stratification at 4 degrees C that we're able to break dormancy. So you can see a real seasonal cycle. I just love this because it really kind of shows how the dormancy can change over time. And it's really important to be aware of this when you're working with seeds, because if you're just playing with seeds and collecting them and sowing them, don't give up on them right away if nothing happens, because it could be if they go through a year cycle, you're going to get to a period in which you've broken dormancy with your seeds. The other thing to see is that over time, in fact, you even get in the second cycle of this species, you even get higher, a little bit higher germination rates. So more of the seeds have broken dormancy that second year and are ready for germination. So we do germination testing at the Miller Seed Vault of all our seeds. And we are trying to not only figure out what kind of dormancy these species have and how to propagate them in the event that we'll need to grow some out, but we also want to know just generally how viable our collections are that we made in the field. So we are taking very limited sets of seeds. You know, usually we're talking about lots of 20 to 40 seeds, and we use these incubators to run germination tests. And the great thing about these incubators is we can control temperature and amount of light. So I have about 4 of these incubators, and I can set some up to be cold stratification, 5 degrees C, or I can set them up to be more like a summer condition at, say, 24 degrees Celsius during the day and 14 Celsius at night and 14 hours of daylight, whereas in the winter, what I call winter cold stratification, I might only have 10 hours of light. And because I'm working with such small seed lots, I use Petri dishes and filter paper or germination paper to do our tests. So we can actually see each week, we log and look at how many seeds have germinated. And then we can also inspect the seedlings and see if they have normal radical development, root radical, and normal shoot development. And then we can transplant them from this tissue paper into a pot to grow them out, which is what you see here. So let me show you how we do our test. We use move-along experiments, which were developed by Baskin and Baskin. And we set up 4 treatments. We have a winter control, which is this blue line, which means these seeds just stay in the winter conditions of 5 degrees C. We have a summer control, in which the seeds, this is the red line at the top, and time, number of weeks is along the x-axis. So these seeds stay in summer for the whole test period. But we have 2 move-alongs, which are the green and the yellow light. So, or yellow line. The green line, which is the winter move-along, we start at the winter conditions for 8 to 12 weeks. We go up to what we have set, another incubator that we set at spring conditions, which is 15 degrees Celsius and 8 degrees at night with 12 hours of light. Hold them there for about 4 weeks. And then we move them to summer and leave them in summer for 8 weeks, move them back to fall, which is the same as spring, and then move them down to winter. And we can repeat that cycle, that seasonal cycle, as many times as we want until we think germination is done. So let me show you a couple of examples of the kind of information we get out of these. So this is a test that we ran years ago on ballhead sandwort, which is Aramagoni ingesta. And this is a species that occurs from the shrub steppe up into the alpine area. And we actually had 5 treatments in this situation. So we also had a spring control, meaning we left seeds just in the spring incubator, which is this purple line. So the green line is the winter control, and that was our best result. So on the x-axis, we have, again, time in weeks. And on the y-axis, we have percent germination. So we got the, and it's cumulative. So the green, we had the best germination of about 92% over the course of this test. But we also, in the winter move along, also got very good germination by the very end of the test at 90%. But what is really nice about these experiments is they give you a little bit more information about exactly what the dormancy is on these species. So for instance, our winter, on our winter control, holding the seeds at 5 degrees Celsius, we see a really significant jump in germination starting in about week 11, and that's about here. And then you see a very steep part of the curve up to about week 19, and then it levels out. So these seeds need about 12 weeks of cold stratification to break dormancy in almost all the seeds. So some were germinating early on, but really by the time we got to week 12 or 13, we really stimulated germination in all of them. But you can see from the winter move along that we actually, in this test, gave them 8 weeks of cold stratification. And at 8 weeks, we moved it to spring. And at spring, you see a huge response. So again, that's conditional dormancy. So these seeds would not have germinated yet if they had stayed in winter, which is what we see on the green line. But when we gave them more favorable conditions, which was spring, warmer temperatures, it triggered a bunch of them to germinate. And so then we see some germination response up to about 75%. And when we took them through a whole nother cycle or series of seasons, we in fact could stimulate more to germinate. So we have a really good picture now of how dormancy worked in this, and I will note, this particular population, which was shrub steppe. Now I said this species actually can also occur in alpine. I wouldn't be surprised if those alpine populations had a very different germination curve. So it would be interesting to compare them. Let me just give you another really interesting example. This is cutleaf synthetis. It's an endemic plant to the Olympic Mountains, and it is found right on the top of the ridges in the Olympics. So keep that in mind, because the ridge tops in the Olympics start to melt off and lose their snow in maybe mid to late May, where most of the valleys, lower areas in the Olympics are still really snowed in. So to see this plant in bloom, you need to be hiking on snow to get up to the alpine. What's really interesting about this species, we ran 4 treatments here. We have weeks on the x-axis and germination on the y-axis, percent germination. But we saw the best germination response and the fastest in the summer move along, which is the yellow line. So we gave it 12 weeks of warm stratification. Summer move along starts in the summer incubator, moved it to spring or fall, and we see a slight response on the graph, put it into winter, nothing. And then as soon as we moved it to spring, we had 100% germination. Conversely, let's look at the winter start, and that's the green line. So we started in winter, had 12 weeks in winter, moved it to spring, nothing happens. Move it to summer, nothing happens. Fall, winter, still nothing happens. And then spring, 100% germination. So that tells us this species needs warm stratification before cold stratification. It's really important. Now, again, this is a plant that has an overwintering bud, flower bud. It blooms as soon as the snow is melting. The seeds are mature and dispersing by mid-July. So it is getting a nice warm, cold, wet, cold stratification, a warm, wet stratification in August, September, and then it goes into winter and then has the cold stratification. So it's a really interesting parallel to the ecological site in which, and the way they have these overwintering buds. This could have really important implications for how this species might respond to climate change. So if we have reduced snowpacks, if we have drier summers, how is that going to affect germination of these seeds over the long haul? So these are the kind of the questions that we can ask once we have this information. Let me just wrap up. I know we're running out of time. Let me just wrap up with a few tips for you if you're interested in trying to propagate native plants. The first I would say is do no harm. Please don't collect from sensitive species, and you can go to the Natural Heritage Program website and they have a list of vascular plants that are considered rare in Washington State. Please don't collect from those species since we're trying to conserve them. And then we have good rule of thumb here is never collect more than 10% of the seeds that are available on the day you make your collection from the population, and don't collect more than 10% of the seeds from any single plant. And this will really protect those wild populations to make sure you're not impacting the next generation of plants. Make sure you also, if you're collecting on public lands, make sure you know what permits you might need to be making those collections. Some public lands require permits, some do not. And then once you've made your collection, you're going to want to dry your seeds pretty quickly to a water content of at least 12% or lower. Slow drying is not great for orthodox seeds. You want to dry them quickly. You guys are really fortunate in Tri-Cities. You have pretty low humidity, but if you're concerned or it's wetter in the winter, you can use a desiccant like silica to dry them out. Store your seeds in a very cool area, out of direct sunlight, good ventilation, in breathable containers. If you really dry them well, then you can freeze them, but you really want to make sure they're adequately dried, because if there's too much water in the seeds, the expansion of the water during freezing will kill your embryos. When you go to propagate them, you can either just toss them out in some soil and see what happens, or you could do a little research. This is a database that we have called the Native Plant Network. Is great because a lot of native plant propagators are putting their protocols into this database, so you can check that. And then also think about what the ecological setting for this species is when it naturally releases its seeds, and that may give you some hints about what is needed. You will want to test to see if the seeds imbibe water. So if you're working with legumes, for instance, if you collect some lupin seeds, you might want to scarify, nick those coats to allow water into the embryo to get them to germinate. So you'll need to think about physical dormancy a bit, but you can then just go ahead and sow them. When I've forgotten about seeds and put them on a shelf and then remembered in December or January, it's too late to sow them outside. So I will sometimes put them on paper towels and put them in a Ziploc and put them in my refrigerator for 8 or 10 weeks. And that gives them cold stratification. And then you can take them out, see if the seeds have germinated, and then sow them into your soil flats. If you're right on top of things, as soon as you collect them, you can just sow them in some potting soil and put them out and just water them occasionally, let them go over the winter, see what happens. And next spring you might have germination. If you don't have germination yet, leave them there for the season. Send them through a whole nother cycle of season, water them, get them go through another winter, and see if the second year of winter temperatures will break the dormancy. So don't give up on the seeds. So I think I'm done. What we can do is take questions. I haven't been paying attention to the chat box. I don't know if we have some questions, but I'm happy to answer questions.
There's a couple questions from Mickey and Jenny in the chat box. Do you guys want to just ask them now, or did you get your questions answered during the talk, or would you like to ask away?
Okay, I see Mickey's question. Can you freeze orthodox seeds to keep them longer? Absolutely. Make sure you dry them sufficiently. So as long as you dry them, get the water content below about 12%. I would use silica gel just to be on the safe side to dry them out, and then you can freeze them. Mm-hmm.
Okay, thank you.
Yeah. And yes, the freezer is in the Miller Seed Vault. So the Seed Vault itself is a climate control. It's got actually 2 climate controlled rooms, And then the freezer is actually within that. So we're actually very strict with how we handle our seeds because we're working with rare plants. So once seeds go into the Miller seed vault, into a climate controlled conditions, we never take them out unless we're going to use them at that point. We dry them, and then keeping the water content low in the seeds is the absolute most important thing for seed longevity. You can vary temperature some, that's okay, but you really want to keep them dry once you dry them until you're gonna use them. So we are always keep them in very dry conditions when we're handling our seeds. Yeah, Mickey.
How long do you keep them? Does that just vary species by species? How long you can keep them in the freezer?
Yeah. So we've pulled out seeds that we've had now for 20 years and are getting as good of germination as we did when we put them in for some species. And then for other species, so what I'll say is that the deterioration curve is, what you see is that it's generally very flat for a long period of time, and then it drops off. So you have, like, if you're testing your seeds periodically, you get, very high viability, and that doesn't change for years and years and years and years. And then all of a sudden you hit a threshold, and all of a sudden you start losing the viability of your seeds. So with conservation collections, it's really hard to know when that threshold is reached, and we don't have a whole lot of seeds to know, to keep on testing. Ideally you might test every year, pull out 100 seeds and see how they're doing. Well, some of our collections are only 500 seeds, and so even pulling out 5 seeds is a pretty big impact to the collection. So we typically don't test regularly. We test at the beginning, and then our collection only dates back about 18 or 19 years. And so we haven't started retesting our seeds. But I know that the Ray Selling Seed Bank in Portland State University in Oregon is one of the older seed banks. They have seeds that have been stored for about 30 or 40 years. And they were testing some of those old collections and were finding excellent viability. So these collections are probably, for most species, are probably centuries. We'll get a very long time out of them.
Okay.
Well, thank you all. I don't know if there's any other questions, but I really enjoyed talking about one of my favorite subjects. So thanks for listening. Good to see everybody.
Thank you.
It was great, Wendy.
Thank you.
I just have one more question then real quick. Do you think you're going to be able to do rare care surveys next year? Do you have any idea?
Yeah, we did them this year.
Did you?
Yeah. So we have a health and safety plan that our volunteers have to follow, of course. We weren't able to go to all the properties. So DNR just didn't open their lands for volunteers until pretty late in the season, like August. But some of the federal lands were open from the start. So we had people out monitoring and seed collecting this summer, and I expect we'll do the same thing next summer, but we will not be having group activities until things change. Yeah.
Okay. Well, good.
Yeah. So take care, everybody. Stay healthy, stay safe, and get outside and enjoy our beautiful state.
Yeah. Thank you.
Yeah.
Thanks
Thank
so
you.
much, Wendy. Really appreciate it.
Yep.
And thanks to everybody that joined. Denise, you may want to stop recording.
I will stop recording. And Wendy, thank you so much. I was the